Hello students and welcome back to Microbial Concepts. So this is another video where we are going to discuss about difference between introns and exons. Now many students they get confused between both these terms. We study introns and exons in molecular biology or in biochemistry where we deal with mRNA processing. Okay, so you can see here already a video is uploaded on mRNA processing if you are struggling with that topic, so you can watch that video. So here we are just going to focus on the difference between introns and exons. So after transcription in eukaryotes, a primary RNA transcript is produced, okay, or synthesized. That is nothing but raw RNA which has both introns and exons. Now this RNA has to be processed to remove introns. Okay, in RNA splicing we remove introns and not exons. Okay, so this is needed, thus it is needed or it needs processing to become a mature one. So primary RNA transcript is raw. After processing, it becomes a mature one which is further used for translation. Okay, so introns they are junkies and exons they are useful. Okay, now what is the difference between both these? So just remember that introns they are interrupting sequences, they don't have any function, so they are removed. And exons they are useful as they belong to coding DNA, okay, or the sequence that codes for proteins. So that's the difference. So first we will see about introns here. So introns are DNA segments which do not code or do not encode for any amino acid sequence in the coding region. And in case of exons it is different. As you can see, exons are the DNA segments which encode the part of amino acid sequence of a complete protein. Okay, so there is contrast. Now introns they belong to non-coding DNA and exons belong to coding DNA. Introns they are considered as bases located between two exons. Okay, interrupt, or you can say they interrupt the sequence of two exons or two coding regions. Exons they are considered as the bases which encode an amino acid sequence of a protein. Introns they are found in eukaryotes, exons they are found in both prokaryotes and eukaryotes. Then introns they stay in the nucleus by splicing out from the mRNA primary transcript during mRNA processing inside nucleus. So if this part is not clear, then you can watch my another video of mRNA processing in eukaryotes. Okay. Exons they leave the nucleus to the cytoplasm after producing or after production of mature mRNA for translation. Introns found in DNA and mRNA primary transcript. Exons they are found in both DNA and mRNA. The introns, the sequence are less conserved, and in case of exons, the sequences are highly conserved. Okay, so this was a short video on difference between introns and exons. I hope the concept is clear to you all. Just remember that introns they interrupt the sequence of exons. So and introns means interrupting sequences, so they are removed, and exons are one which are useful, which encodes for proteins. Okay, so I hope the concept is clear here. So do like my videos, do share my videos with your friends, and do subscribe to my channel.